So today I would like to share, first I would like the meeting organizer for the invitation. And it's my great pleasure to share you our current work on watermelon cerebral pine genome. And watermelon belongs to the Citrulus genus. The genus currently all includes like 7 extant species. The first one is, the pointer is not moving that smoothly. The Lanatas, and that's including the cultivated watermelon, and also a wild form like called Phanos. And we have Mycosopomos, Amaras, Roses. I'm sorry. Oh, maybe here, right? No. Okay. It's the Roses and Rimii, Collisensis, and Nodiananos. Okay. So the 3 are highlighted in red, like Mycosopomos, Amaras, and Collisensis. That has been used in the breeding program. The other 3 wild species has barely touched because we don't have enough germplasm available. So first, I would like to introduce that during the past 15 years, the genomic resource we have developed for watermelon. So in 2008, led by Dr. Yong Xu, we formed the Watermelon Genome Initiative. And our goal is to generate the first reference genome for watermelon. So in 2013, we published the first watermelon reference genome, but this watermelon genome was sequenced using the short-read technology, the Illumina technology. And then in 2019, we used the long-read technology to improve the reference genome. So then, in collaboration with Amnon, we generate another reference genome for cultivated watermelon. That's the Charleston Gray. It's an American ecotype. And you can clearly see the difference between these 2 types of watermelon. One is round, one is oval. The fruit shape is different, and the fruit weight is also highly different. So during the past several years, we developed several reference genomes for the wild watermelon species. So the first one is in collaboration with Susan Renner, we developed a reference genome for Phanos. So this one was assembled using long reads, and we provide enough evidence to support that this one is the direct progenitor of cultivated watermelon. Then in collaboration with Shekel, with Pat, and Amnon, we developed the reference genome for other 3 species that are widely used in the breeding program, the Mycosopomos, Amaras, and the Collisensis. So besides the reference genome, we also did a lot of genome resequencing. So in 2019, we published a paper describing the genome resequencing of 414 watermelon accessions, and which include accessions from all the 7 extant species. So here is the list. Here's the number of accessions in each species. And then during the past couple of years, in collaboration with Amnon, with Pat, with Sandra, and also Shekel, we did additional genome resequencing for this wild species. And we also, I wanted to mention or emphasize that in collaboration with Cecilia, we generated genome resequencing for the Phanos, 15 accessions that were provided by Cecilia. And this one kind of like fill a gap. So previously we have very few, 2 or 3 accessions belong to this direct progenitor. Now with this 15 newly sequenced Phanos, we can revisit the watermelon domestication that I will talk about in the next few slides. And this is the final a summary or integration of all the genome resequence data we have generated. So in total, we generated about 447 genome resequence for 447 watermelon accessions. So this will provide a solid basis for the pangenome construction. But before we go to pangenome construction, as I just mentioned, we want to revisit the watermelon domestication using the Kordofan melon as the direct progenitor. That's, as I mentioned, we provide evidence to support that this is exactly the direct progenitor. So previously, we used Mycosopomos as a direct progenitor, but that turns out to be, you may lose a lot of information using that as a direct progenitor. So you can see here, we grow these 15 codofanous accessions that are provided by, the seeds was provided by Cecilia in our greenhouse. And you can see the phenotypical diversity of these 15 watermelons. And the take-home message here is that all these codofan melons are non-sweet. So my student, Hong He, in the audience, tasted all 15 watermelons. This is non-sweet. And also, I'm sorry? Oh, okay. So they are non-sweet, and also they are non-bitter. You can see red or pink colored fleshed watermelon in codofan melon group. And we did genome resequencing of all these 15 accessions, and we performed, we call SNPs and generate a phylogenetic tree of that 547 watermelon accessions. And we found that this is all these 15 watermelons, codofan melons, and the other 3 that sequenced previously, these 18, they clustered into one monophilic clade, and very close to the domesticated watermelon. This provides additional evidence to support this is an independent group, and could be the potential direct progenitor of watermelon, of cultivated watermelon. So then we compared the genome of the codofan melon and the cultivated watermelon, the landrace, and we identified selection swaps, and we found that these selection swaps overlap with a couple of fruit sweetness QTLs. That means during the watermelon domestication, you can see from codofan watermelon, sorry, does not work. Okay, so from the codofan melon to landrace, then to cultivar, there's a huge increase in the sweetness, the sugar content. So the sugar content could be the most domesticated trait in watermelon. And that's why we got a lot of domestication sweep overlapping with fruit sweetness QTL, both during the domestication and also during the improvement. Oh, sorry. Okay, hey? Confused. Okay, so then the fruit bitterness. As I mentioned, the fruit, all the codofan melons, they are non-bitter. And that means that the non-bitterness trait had already been fixed in the wild progenitor. And it is not a domestication trait. As we always think about bitterness is the domestication trait of watermelon, but it turns out it's not. It's already fixed in their direct progenitor. And we checked the nucleotide diversity between different groups, like Mycosopomos, and landrace, codofan melon, and cultivar. And we found out that in codofan melon, their genetic diversity, nuclear diversity, is about the same as the cultivated watermelon. That provides another evidence that this region is not selected during domestication. It's already fixed. Then the fruit color, as I mentioned, we already have the red color in the codified melon. Not like previously people think about that the all codified melon we have, like the 3 codified melon we previously sequenced, they are white flashed. So the people conclude previously that the red color occurred in the early stage of domestication. But that's not the case. We found out that this red flashed color is already in the progenitor, and the red flashed color could be used as a starting material for domestication. And we also found out that this gene, lecpin beta cyclase, and this gene was under domestication, and this is the gene controls the fruit color of watermelon based on the nucleotide diversity. So now we back to the watermelon pangenome. So we, as I mentioned, we generate enough and a huge amount of reference genome and genome resequence data. And you can see these 2 pictures actually was provided by Xinping Zhang. And you can see the phenotypic diversity of watermelon. That means that a single reference genome or a couple reference genome cannot capture the genetic diversity among the population, because they have huge variations. So that's why we need pangenome. So first we build a pangenome using the map-to-pan approach for each watermelon species, and the 4 species, and the other 3 species because we don't have enough accession to work with. So we built pangenome for each of the 4 species, and we got from 2000, we got 2,200 genes, novel genes for cultivated watermelon and the codified watermelon, this Lanatas species. And we got 583 for Myxospomos, and close to 2,000 for Amaras, and 2,500 for Colicensis. So these are novel sequences and provide a lot of information. And then we caught a gene presence and absence variation. We identify those genes in different categories. The first one is in pangenome, we call it core gene. That means the genes that are present in all accessions in the population. And you can see that in, I'm sorry, I can't use this pointer. Okay, you can see that the core gene in these 4 different species are quite high, especially in Myxospomos. It's 97.2%. That most of the genes are present in all Myxospomos accessions. So yeah, that means the Myxospomos, they have, the genetic diversity is higher, actually is higher than Lanatas, but their core gene content is higher than Lanatas, also higher than Lanatas. That means the gene presence absence variation or gene loss or gain among the Myxospomos population is real. It's less real, is less often than the Lanatas or other 2 species. Okay, with this 4 reference genome, 4 pangenomes, now we build a watermelon super pangenome by integrating these 4 pangenomes, the pangenome for the 4 species. And we use an approach that using the gene leftover among the 4 species, and then to establish the orthologous relationships. And we also use the syntactic orthologous genomic regions and to identify syntactic blocks. So in total, we identified about 38,000 orthologous gene groups, and most of them they are syntactic orthologous groups, and we identified about 3,000 singletons. That means that's one gene in one specific species, the species-specific singletons. And the take-home message from this analysis is that we found a lot of orthologous genes contains predicted protein coding genes in one species, but they have matched location in other species, but there's no gene can be predicted. That means in that region of that species, this gene could be pseudogene, or the function could be lost. And as shown in this figure, you can see the first one is gene to protein coding gene, and also gene to location that does not contain predicted genes. And we got like 1 to 1 to 1 is 28,000 genes, the last column. But if you only consider gene to gene, predict gene to predict gene, that number down to like 16,000. You have a huge decrease. That means there's a lot of genes that are present in the genome, but their function could be lost due to near functionalization or pseudo generalization. So this shows the core gene content of the super pangenome among the 4 species. So the super pangenome, the core gene content is 63%. It's much less than the species-specific pangenome. So this indicates that there are diverse genetic makeup among the 4 species. Right? The difference is mainly from between species. Within species, the difference is much, much smaller. So I expect the core gene are expressed in higher levels. And also, when you check the gene content in each species or each groups, we found that gene loss during watermelon domestication. So this codophilus to lanolase to cultivar, you can see there's a decreased pattern. That's also found in tomato, the gene loss during watermelon domestication. And also that when you consider the gene functional, the predicted gene, the cholesynthesis has the highest gene content. But when you also include the gene locations, then cholesynthesis has much less gene content. That means that different species retain different number of functional genes. And in cholesynthesis, they retain much more functional genes, but in other species, they retain all the gene was mutated, become non-functional. So we identified, we checked the gene absence and presence variations, and identified the select genes. That means genes that have different occurrence frequency in different groups. We identified a huge amount of genes between different species. But during domestication, we only identified very few genes that have under selection. This is not unexpected because we have a very short history of domestication, like several thousand years. I'm giving a couple examples. I know I'm almost out of time, so I will go faster. So this is at least the frequency. That Y means 100% in those accessions, in the accessions of one group. And you can see these 18 disease-resistant genes were all either totally absent in cultivar watermelon or have very low frequency in cultivar watermelon. But they have very, very high frequency in at least one of these wild species. So this provides very, very valuable material for future breeding. And also this TS2 genes, and that's also reported in chimpanzees G42 genome. We found 2 copies based on the orthological relationship. We found 2 copies of TS2 in cultivar watermelon, but only 1 copy in wild species. And in codophilus, there's only 1 accession containing 2 copies. And interestingly, this one has the highest sugar content. So TH2 was involved in sugar content, in regulating sugar content in watermelon. And we also found that in cultivar and landrace, accessions with 2 copies has much higher sugar content than accessions with only 1 copy. And also this gene is under selection based on the nucleotide diversity. And the next, I will spend like 1 or 2 minutes talking about the current work on graph-based pangenome. So I will ignore this slide that just tell you how good graph-based pangenome is. But we need to have a high quality reference genome for multiple accessions. So this is the last slide I'm going to show. This is we already generated reference genome for 139 watermelon accessions, and among which 135 was newly generated, and 4 of them that in blue generated previously, including the G42 Xinping generated, and also these are all long-read sequence, and we can see all this genome was assembled in very, very high quality. Most of the chromosomes are assembled in 1 or 2 contiguous. So we are in the process of constructing this graph-based pangenome, then integrating with this genome resequence data. I believe like in the next year, we can give you a much better story on watermelon domestication and evolution, and also the important agronomic traits. So with that, I would like to thank the people in my lab and also our wonderful collaborators and funding agency. Thank you very much. Yeah.
Any questions?
I may take one question if you have. I'm sorry, can you? Oh, your sugar content and disease resistance. Yeah. So that's a very good question. I don't think they are highly correlated. Yeah, but I mean, that's in tomato, some tomato flavor gene or fruit size gene was negatively correlated with fruit weight. But I'm not sure for the watermelon, the sugar content and the disease resistance are highly correlated. Maybe some breeding guy, maybe you guys can provide a better answer. I don't think they have huge correlation.
Yeah. The more sugar, the less resistance.
The answer is no, right? Yeah.
No,
That's also my answer. Thank you.
Catherine, did you want to ask?
Thank you for your presentation. Can you mention the origin of this material at the geographical point of view? The name is Kordofan. Kordofan is in Sudan, but I don't know if the collection, the accession you studied, do you know their geographical origin?
Yeah, the geographical origin is from Sudan, the Kordofan region. Yeah. So actually, I think Susan Reno got the seeds from some guy from Russia, collect from Sudan. Then they ship seeds to me and also to Cecilia. Then Cecilia, I'm not sure how many generations you saved. One generation, we saved 1 generation, then I got that 15 grow in our greenhouse and did the sequence. Yeah, thank you.
Harry, one more question.
Oh, okay.
Harry Paris.
Zhangjun, how certain can we be that the hundreds of accessions that you used, Citrullus is notoriously, the species cross-pollinate and interact with one another genetically, notoriously. How can we be sure for each species or for each accession that you used that they are purely, say, 100% citron watermelon and didn't interact with dessert watermelon or Mucosospermus, and vice versa for the various taxonomic layers you have there?
Yeah, yes, that's a great question, So first, we use our evidence to use genetic analysis like a phylogenetic tree, and also the population structure analysis. And we can find there, actually the slide shows there, most of them, the background are pretty pure. But we did identify some integrations from wild species to the cultivar. That's the effort of modern breeding program. And we did find that integrated region contains disease-resistant genes. But I believe most of the exceptions, maybe 99%, they are highly consistent with their taxonomy. And we did remove some exceptions that have inconsistent taxonomy. That's in our study, we did remove those. Yeah, thank you.